Okay, so I had uploaded I don't want to get too complicated, but I had uploaded this to a Docker image so that you guys would be able to run it and also edit it in real time, but it doesn't like the library because I think I needed to install it some other way first. So we'll just walk through the code. And if you want to run it yourself after we do the activity, I'll troubleshoot how to get it to work on that other thing. Otherwise, this code will also be available in the same place where you had access to those files. So you could always copy and paste it on your own to a Jupyter Lab environment and play around with it. So the first thing that I wanted to go over was, so we had this data file, and I'd said there were different files in here. So I wanted to just quickly describe the files. Daniel, did you have a question? Or no? Okay. So this file, the TCGA driver genes file, so this is a list of all of the most common cancer genes that this study called the Cancer Genome Atlas Project identified. And I think this is about 20 different types of cancer. And this is actual data from the scientific study. I didn't do anything other than I just removed a couple columns that weren't necessary. And I wanted to just kind of describe it to you so you can see what information is here. And you can also use it for other things if you kind of want to explore more about cancer genes and their association to different cancers. So on the left, in this first column, you have the gene name. And so I believe you guys went over this when you learned about OMIM and the gene symbols. Like you can just search those and it'll tell you all the information that we know about that gene. You can search it in Google, you can search it in Wikipedia, you can search it in OMIM or like any other place you think you can find information about a gene. And then the next column has the tissue type for that cancer, which is the third column. So the actual full name of the cancer is given. And one of the things you can notice is if you kind of scroll down, you see a lot of these are carcinomas. You know, the name's carcinoma pops up a lot. And if you go back to what we talked about in the first couple of slides about like the naming of tumors, and that 80% of tumors are carcinomas. So here you can see like it's actually in the name carcinoma. And then the last column has tissue frequency. So what this is it's a decimal that if you multiply it by 100 is the percent, okay? So this is the frequency of that gene being mutated in that type of cancer. So this ATRX gene is mutated about 4% of the time in the adrenal gland or adrenocortical carcinomas. Or ATM right here in bladder cancer is mutated about 13% of the time. So what this will tell us is how frequently a gene is mutated in the cancer and how important that gene is to that cancer. So if the gene has a high tissue frequency, that means that cancer gene is very, 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 very common for that cancer, which means it's very important for causing that cancer. So I want you to have that kind of mindset. Some of these tissue frequencies are really high. So I can find one if we scroll down. Here we go. The TP53, so that's actually the most commonly mutated cancer gene, period. About half of all cancers have a mutation have a mutated P53 gene. And in bladder cancer, it's about half, 48%. So some of these are much more common than others, like AKT1 in breast cancer is only 3%, okay? And then I'll go over one sample file so you can kind of see the structure. So this is a table of mutations. So like I said, this is actually from a real patient. I just took a subset of mutations from a TCGA data set. So this same big study of cancer, but you guys don't know what type of cancer this is. You don't know anything about it other than there's this mutation. And then I've listed the genes. So we have the chromosome that the mutation's on, the specific base pair position, the original base letter, sequence letter, and then this should be ref alt. That's not right. And then what it's mutated to. And then the last column are the gene names. So the most important thing here, like I said, is just remember that each row is unique to a mutation. So there won't be anything in common between the rows. There might be multiple genes of the same name that have different mutations, but the position is going to be different. Okay, so with that said, we're gonna go to the Jupyter Notebook, and I did upload this to the GitHub, and I'm going to send you guys the link of where you can see the code and follow along yourself. I wish I had the thing working where you could run it. It's just, it's not working now. So it's right here, if you want to go there. So this first section, what we're going to do is we import this one library, and this is all we need. So in Python, a library is basically a group of tools that help you, or it's a group of like little pieces of code that you can reuse to do analysis without having to write all of that code again. So this library called Pandas has a bunch of pieces of code that help you organize and manipulate data. And it's my favorite package to use. I use it all the time. So if you do want to learn Python, or if you've already started to learn Python, definitely learn Pandas and learn the different things you can do there. So for this first line, what we do, or this first chunk, we open that library of code. And then what we're also going to do is we're going to read in our data files. So there's a function in Pandas called read CSV. So all of these files are CSV files, and CSV file just means that it has a comma between each data. So it's comma separated variables, and that's just a file type. So reads it in, it stores it in these different variables so we can work with that data. So I'll run that. And so it ran it. I'm not printing anything to screen, so there's nothing to show for that part. So once we have our data in, we kind of need to kind of organize it in a way that we can work with it. So right now, first thing we want to do is we want to make one big table that has all of the samples together, rather than the samples being in different files, because our goal is to identify how these samples are evolutionary related, which one came first, second, third, last, so on and so forth. So in order to see those patterns, it's going to be so much easier if we have it all together. So the first thing we want to do is we want to add a column to each sample file that has a sample ID in it. So when we join them together, we know like which row came from which sample. And then we're just adding a value placeholder to show like that sample is for that mutation, and it'll make sense when we get to the next stage. So then the next stage is our final table that's going to join everything is going to have all of the mutations that we have in total on, like, as the rows, and then as the columns, we're going to have the information about those mutations, and then sample whether or not true or false if they are in sample A, true or false if they're in sample B, and so on and so forth, all the way to E as columns. And you'll get to see what that looks like in the last step. Oh no. Oh, sorry, this piece of line needs to go there. Go above it. This is also why it's important to run your code in full, not in parts, because I read it in parts before you share it with people, so I apologize. Okay, so then we've kind of like done this cleaning of the data. And so even though I'm doing this in coding, you can actually do this stuff by hand. And that was one of the reasons why I gave you a smaller data set, so that you can take the code and then do it by hand and see like how, if I merged all these rows by hand, like I literally copied and pasted everything, this is how the code's doing it. The other thing about my code, I didn't use any big loops because I wanted you to see each step very clearly. So you can really optimize this, like this section, for example, with a loop where it has, it goes through each sample ID, goes through A, B, C, D, E in a loop, rather than I'm running each one separate. That's a better way of writing the code, but I'm trying to show you explicitly the code. So I didn't want to like make it more complicated than it needed to be. Okay, so then we're going to group all the samples into one table, and we're just gonna now do what's called a pivot table. So it basically just takes some of the rows and makes them a column. And what we will also do is I'm going to add another thing so I can show you what it looks like. So sample A doesn't have that. Okay, so I just added two little pieces of code just so I can show you what the data is looking like as you're seeing it, as we're working with it. So this was how it looked when we started, minus the sample and the value part. We had the chromosome, the gene name, the position, the alt and ref columns. They got resorted when it reads it into Pandas, so just ignore that the columns are in different positions. And then we had added that sample column, and then we'd added this value column in those first couple steps. And then what we did was we added up, basically we glued up all of those samples, those 5 different samples together into one table. So now we have the chromosome information in the gene. So we have the mutation information in the gene, and then we also have all of the different samples, so A, B, C, D, and E, listed as columns with a true, or it should be false, but it has non in there. It decided to use that instead of false. So true means that that mutation is in that sample. So this mutation is in sample E, and it's in sample A. Anything else means it's not in that sample. This mutation is ROR1 is in sample C. This LELP1 is in all of them, and so is the other two. So this information is going to tell us now, okay, we can look at this table and say, these mutations are all in this sample. This one's in this one. This one's in this one. What's going on here? We can start to see what the pattern is. The next thing we want to do is we want to start working on that cancer gene file and try to pull out some information we want from there. So one of the things we can do is we can pull out the cancer genes that are in our sample files. So just because there's a mutation in the tumor, it doesn't mean that that mutation is in a cancer, like a really important gene. We want to pull those ones out because that's going to give us way more information than these other mutations. So we'll run this code. So what this does is it pulls out the genes that are in our sample table, and they're in our what we call our drivers list. So there are, and then we're printing the list right here. So there's 5 genes. There's KRAS, TP53, SMAD4, FBXW7, and APC. So those 5 genes, those are the only cancer genes we have in this patient, which it statistically is actually about right. Yeah, that's about what we would expect. We usually see like a dozen or less known cancer genes in a patient, but we might see hundreds of mutations or even thousands in the cancer patient or the tumor. Now what we want to do is we want to find out more about these cancer genes. So now we're going to start to figure out what type of cancer is this sample, because now we know there are these genes in it, and that other file gave us information about those genes, how frequent they are in certain tumor types. So we want to basically add that information, annotate that information onto this list of cancer genes. So that's what this little line of code is going to do. And we will print it too. All right, so then what we can also see, and I think I have this printed, actually I didn't print it, it was supposed to be printed, but we can actually see here that P53 is in the adrenal gland tumors at 17%, and a few of these other ones are in bladder, and this is in breast. So this table is not the full table of all the data. What this command does is it just prints the first couple lines. We want to see all of it because, or actually, no, that's the next step. Sorry, I'm getting ahead. So now what we want to do is we want to see, we actually want to see what genes are high, which of these genes, which ones have a high tissue frequency. So what we're going to do is we want to sort by the tissue frequency. So that's all of this code. So we're going to sort the whole thing with this first line. And then for each gene, each of those 5 genes, we're going to sort and print so that you can see what it looks like. Oh, I have to do this separately.
Is all of this work usually put into making a table?
Usually when you run this work, if you're, if you do this every day, we wouldn't print all of the middle steps. We would just run the code fully through. And then at the end, we would just save the last few data files, but because I'm trying to show you like how the data is changing, I'm printing kind of at every step so you can see the tables. But that's why Jupyter notebooks are really good, because you can do this and keep it all in one document. Okay, so then I'll scroll back up. So the first gene we looked at was APC. And so now you can see the top cancer that APC is associated with is what? Colon cancer. Colorectum is the tissue, colon adenocarcinoma, and it's 81% of those tumors, right? The next cancer is stomach, and it's about 9%. And then after that, it's prostate, and it's about 2%. So given that our sample has APC, it's it's looking like, okay, it could be colon cancer because it's 82%. But let's look at the other genes. If we look at KRAS, which is the next gene we saw, it's found in 73% of pancreatic cancers, and it's found in 47% of colon cancers. So now it's looking like, well, okay, maybe it could be pancreatic cancer, but it's definitely probably colon or pancreatic, given what we're now seeing from just these two genes. And if we look at SMAD4, we're again seeing pancreatic and colon cancer at the top. Again, pancreatic has a 23% frequency, colorectal cancer has 12%. Now when we look at P53, it gets crazy, because remember I said P53 is the most common cancer gene in all cancers, and it's found to be mutated in like half of all cancers. So actually 92% of uterine cancers have the P53 gene mutated. And then you have ovarian. And then when you get down the list, we see colorectal cancer is 72%. So now that one kind of, raises a question. But then the last gene, when we look at it again, we see uterine cancer, and colorectal cancer. So none of this is a definitive answer, but if looking at some of this data, what is your hunch telling you about what type of cancer this is? Does anyone want to take a guess? You can unmute yourself, or you can feel free to throw your answers in the chat. So Leandra's guessing that it might be colon. Skylar says colon because it was listed in every single gene table. Any other guesses? Anyone think it's something else? Anyone think it's not colon cancer? Okay, so we'll come back to that. So those are good guesses. I won't tell you what the answer is until after we're done with everything. So, and the reason why I'm not telling you the answer is because as a scientist, if I was given this data, I wouldn't know the answer. I would have to go off of my best guess based off of the data that I'm seeing. But it's still ultimately a guess. So then the last part that we want to do is now we want to look at the order of these samples, now that we've kind of gotten some more information about what kind of cancer this might be and what cancer genes are present in this cancer. So what we're going to do here is we're going to sort that big data table, and we're just going to sort it by the columns A, B, C, D, and E, and just sort it in order. And then after we sort it, what we're going to do is we're going to only take out the rows where they had a mutation in those 5 cancer genes, so that we don't have to look at all of the data because there's, I think, about 100 mutations in total in that file. And so this will help us be able to figure out the order a little bit more clearly. Okay, so I ran that code, and then what it did was it printed out the mutations that were in either APC, KRAS, SMAD, for P53 or FBXW7. And what you can see is that there's only one mutation in each of those genes. So it kind of makes this a little bit easier. And if you look at APC, it's present in every single sample. So you can see here, it's in A, it's in sample A, it's in sample B, C, D, and E. Okay. KRAS is in all the samples except for B. SMAD4 is in only A and E. P53 is only in sample A. FBXW7 is only in sample C. So I'm going to give you 2 clues to help you think about what the order might be for this. And then I'm going to give you like 5 minutes to kind of wrap your brain around it and maybe like sketch out on paper how you think this might be organized with the order. Like if A, like you want to do like, is it A, B, C, D, like, or is it B, A, D, E? You want to try to sketch out that order of the tumor evolution. So the first part, the earliest stages of tumor evolution, you can pretty much say that those mutations are going to be present in the later stages too. So if you see a mutation that's present in all of the samples, that could suggest it's earlier. Or if you see a mutation that's present in more samples, that could suggest it's an earlier time. If you see that mutation is present one time, it's probably a later one. So think of it like that. The later stages of evolution are going to acquire more mutations overall. So they're also just going to have they tend to have a bigger number of mutations. Earlier stages of mutations of evolution tend to have a fewer number of mutations. So those 2 pieces of information can help you figure out the order and how to order these samples. So I'll give you, like I said, I'll give you 5 minutes. Please throw your questions in the chat box and I can answer them, or you can unmute yourself and ask any questions you might have.
Would that mean that TP53 would be very high since it's in a lot of the different cancers?
Well, so yeah, P53 is found in a lot of the different cancers. But in this example, because remember I gave you one patient's tumor, but we're looking at it across 5 different time points. So we're looking at, like early, middle, and late. If you look at P53, it's only mutated in one sample. It's only mutated in sample A. So it's common because it's kind of inevitable. Like it's going to happen at some point in the tumor, but I don't want to give you the answer, but what do you think? Do you think it's an early event or do you think it happens later? Would anyone like more time, or are you guys ready for some answers? So the question is, using the information down here, let me show you else. Using this table, and I kind of drew over it. Using this table, can you put those samples back in order? So A, B, C, D, E, what is the order of those samples in time? So which one came first, second, third, fourth, fifth? And it's okay if you can't fully figure it out, but maybe you can get one or two of the samples. Maybe you know which ones are early and which ones are later, but you don't know the specific order. That's okay. But try to think about it. So Daniel thinks C is last. If there's any people that agree, you can say I agree, or if you think it's different.
May I have a question?
The question is, what is the order of the samples in the tumor? And you use this table if you're looking at my shared screen that I circled in blue.
Yeah,
So you have,
I heard that. I want to ask about
oh,
the
sorry.
one, the one with much true is the older one. Is the earlier one?
If it says true, it means that there is a mutation in that sample. And if it says NaN, it should say false, but for whatever reason, Python put NaN. That means there's no mutation.
So B gonna be the least cancerous one?
B is showing that it has only a mutation in APC. So it's not necessarily cancerous, but it could, it does mean something. What do you think it means?
Okay, so, okay, it looks like we're getting some answers. Does anyone want more time? Or do you guys want to talk about it? Okay, no one's asking for more time, so we're going to talk about it. So let's talk first about what cancer type this is. So we had some guesses that it was colon, and I think there was one guess that it was urinal, and I don't know how to get this annotation off. Oh, okay, there. Okay, so we had colon or urinal. So I will say this is colon cancer. And so was it Skylar? She made a really good observation that colon cancer, colon adenocarcinoma was the only tumor type that was listed in every one of these tables, which is true. And so it was listed in every one of these tables, and it was also the most frequent for APC was the most frequent for colon adenocarcinoma by far from the other ones. And usually when you see that one gene is really, 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 really common in one cancer type, it means that if you ever see that, it's usually that type of cancer. So usually when you see APC, it's going to be colon cancer. So very good observation, Skylar and Leandra and Daniel, and hopefully you learned a little bit there from like how we kind of go through data and figure out what we're dealing with if there are unknowns. So in terms of the order of the samples, so there's some guesses that B is first. And so when Sharif asked his question, I didn't want to tell you that it was first, but I knew that that was what you were getting to. But yes, B is first because it's the only sample. If you look at the column, if you look at it as a column, the B column, it's the only sample where you have just one mutation. So it means that, and the one mutation that's in that sample is in all of the samples. It's that APC mutation, which means that it's in all the samples. So that has to be common. So it has to have happened early. And then sample B is the only one with that one. So that was the first sample, the first time point. Okay. And then how we would find the next one. So the really simple way of doing this is there's five mutations in 5 samples. So if the first sample if the first time point, the first sample has 1 mutation, how many mutations do you think the second time point will have? We'll have 2. So if we look at the columns again, the next sample that has that number that has 2 mutations is D. So it goes B, then D, and then the next sample should have 3. Now here's where we have a problem. C and E both have 3 mutations. So I gave you a trickier one. So this just means that they're both next, but I'm going to draw it out so you can see it. The order is actually going to be branched. So you'll have a branch like this from D. It'll branch where you have C and E because they both have 3 mutations. And now the last sample, which is A, we have to figure out, does A come after C or does it come after E? Well, A and E have the most mutations in common. So it comes after E. So it's like that. And then at the top, you'll have E, if I can draw it. E. Sorry for my bad sketching. But that's the order of the data set that I gave you. But what I want you to do too, if you want to kind of like take a stab at more of this outside of the and sort it and really look at it, and you'll see this pattern. It's just when you're kind of dealing with it in a coding environment and everything's behind the scenes, it's harder to see, which is why I only pulled out these few mutations. So I hope, yeah, sorry, Eden. Yeah, it's very confusing, very interesting. And the reason why it is very confusing, I'll tell you this. I didn't get into this because I wanted to give you guys a challenge. I didn't want to give you like really easy stuff that wouldn't really kind of work your brain a bit. And I also wanted to give you stuff that we really question and we really work on as cancer researchers. But I actually did my whole PhD in this. I had samples from kidney cancer, actually, and it was patients that had multiple tumors in their lifetime. They had the same type of kidney cancer, but different time points of it. And I reconstructed these trees, and it was a pain, and because I had to do it computationally and a little bit by hand because there wasn't the programs at the time to kind of handle all that data. So I had to kind of write some of my own code to create that analysis. So anyway, that's kind of the backstory of this. So I will update the code on GitHub, the Jupyter notebook that we worked with here. I'll update it so that you can actually have this one. I'll make sure to share it in the Slack page so that you guys can always go to that link and download it if you want. And I'll also throw up the slides that we went over today. And I have just a couple more things that I was going to talk about. Let me pull back up my slides. Does anyone have any questions before I start talking again? Okay.
Why does cancer sometimes come back to people after all of like the treatment, like chemotherapy?
Yeah, that's a really good question, Daniel. So there's a couple reasons. If you were unlucky and you had, well, remember we talked about the germline mutations, the mutations you're born with. If you're unlucky and you had one of those that's cancer causing, you're kind of always going to be at risk for cancer, even if you treat the first one you get. So there's that case. The other case might be that you caught the cancer too late and it spread to other tissues. So now instead of just having the one tumor or the one type of cancer, you have multiple types of cancer in your body. And it's just, it's a natural part of the cancer's evolution to want to get to that stage where it spreads. So the whole point in treating cancer is to catch it before it spreads, before it metastasizes, because once it does that, it's really hard to treat and it's probably going to come back if they do treat it. And then the last way that people can get cancer again after they've had it is from the cancer treatment. So radiation is a common cancer therapy, but radiation also causes mutations. So when we talked about sunlight causing mutations, I'd also showed you that one slide where it briefly mentioned like X-rays and stuff. So every type of radiation will cause mutations. And so there even are some cancer drugs that can cause more mutations. So there's this balance of treating the cancer without creating more cancer, on the back end. And then also just by random chance, you could just unfortunately be lucky, or sorry, unlucky to just unfortunately get cancer another time just by random chance. But it's usually those other three. That's a good question. Any more questions? Okay. So this was just the diagram sketched out neater so you can see. So I wanted to show this figure. So this is The reason why I gave this example of colon cancer and I gave these specific genes and this discussion is because this is one of the best examples of showing how a tumor evolves from a normal cell to a like abnormal group of cells to this weird structure all the way to an invasive tumor that can metastasize and all of that stuff. And this model of how we understand cancer was created in understanding colorectal cancer back in the early 80s, I believe. It might have been the late 70s, sometime around there. So we've known about it for almost 40 years, like that cancer does this particular thing. And so this figure just kind of shows where those cancer genes that I was talking about, where you see the mutations happen and then it causes a change in that tissue structure. So here the colon, change in the colon structure. So you get the APC mutation, and then later you get this KRAS mutation, and it causes what we call polyps. And they grow bigger, and then at the end you get that P53 mutation, and then you end up with full-blown carcinoma. Skip that slide, skip this slide. So I was just going to briefly mention a little bit about my background so you can kind of understand like what I do and why I've been talking to you the last few hours. So I work as a clinical data scientist and also in bioinformatics. And so, as you know, you might have heard of data scientists, and they are people who extract knowledge and insights from various data sets. And bioinformaticians, they specifically work with biological data. So bioinformaticians can really be like biological data scientists. So that's kind of how I like to think of them. And we use a lot of computer science and statistics to understand things about biology that are really complicated. And so computers can help us. And we get data from all sorts of places, whether it be the environment, from clinical settings, from individual cells, from lab experiments, from DNA sequencing, all sorts of places. So, well, let me just go through it because I wasn't supposed to do that. So my path in my career started in high school when I was interested in science, and I decided when I was applying for college that I was going to apply to science programs. And so I applied to study biology and actually marine biology in college. And then I settled on going to the college I graduated from, Chatham University, which was in Pittsburgh, Pennsylvania, which is on the eastern part of the United States, if you're not from the United States. And there I studied biochemistry, and that's where I first learned about bioinformatics. And when I learned about bioinformatics, it was my junior year, because we had to do an undergraduate research project. We had to do an undergraduate thesis. So I know you guys some of you are really close to college years and some are a few years away. But one of the things I would recommend is when you start thinking about where you want to go to continue your studies, think about what opportunities are already available at that university that will help you get to your career easier. So I chose this university because there's two things I wanted to do. I wanted to travel and study abroad, and I did want to do some sort of like research. And so both those two programs were part of my requirements to graduate. So I got to study abroad in Europe, and then I got to in Belgium, and then I got to do my research project. And then after I graduated undergrad, I did a summer internship at the University of Pittsburgh, which is a big university in the same city that I did my undergrad in. And there it's a big research institution. So they had all sorts of scientists doing all sorts of cool things. And I got to work in a lab that was building software to analyze genetic data. And at that time, we couldn't analyze stuff on our own computers. Like the data I was giving you is really small, but the data we would work with would have millions of mutations in each of them, and sometimes like a billion. The files would be that long. So our computers weren't powerful enough to do it. So we had to use big computing networks. And I was very fortunate because in Pittsburgh at that time, they had the second most powerful computer in the United States. And so because we had access to that computing resources, I could do some cool research. So another key piece of information I would give you is that think about the environment that you're going to do your studies in. What other things will you have access to simply because you're in the right place at the right time? So if you do want to continue this study of computational biology or bioinformatics, you definitely want to be in a city and university environment that has access to a lot of computing resources and probably a lot of collaborations with companies that build computers and things like, or that build cloud computing systems, or, the big tech companies like Google or Microsoft or Amazon or Intel or any of those big ones, maybe even NVIDIA. I'm sure they do collaboration. So after that experience, I ended up doing a graduate program in biomedical informatics. But it didn't feel quite right because I wanted to get back to genetics. And the reason being was that I came into genetics because I had a cousin who had a genetic condition and passed away from it. So as I was growing up, it was always something that was interesting to me, but it wasn't until I got hands-on in that kind of field that I got to really see the impact that I could make in that area. So I decided to take a step away from graduate school, from this graduate program, and really focus my training more on learning more about genetics and how we create the data sets that we work with. Because the lab I was working with was all computer scientists, and we were trying to build tools. We weren't trying to create the data, and we weren't trying to work with patients. So I wanted to be on the other side. So after that, I moved to where I live now. I moved to the other side of the country to Oregon, and I got a job as a research assistant at the Oregon National Primate Research Center. And I got to work on pretty cool research where we looked at how traits are passed between families in monkeys. So we worked with rhesus macaque monkeys, which are the ones in the picture I'm showing you. Actually, those are rhesus. And I got to build these really big pedigrees of monkey families, and look at their genetics to see how we could kind of track how certain traits were passed down. And the traits we were interested in were blood lipid levels. So blood lipid levels are things like cholesterol. Like if you hear like, oh, my mom has high cholesterol or things like that, we're interested in looking at those sorts of molecules because that can tell us how common cardiovascular disease might be in families. So that was really cool. And while I was there, I also I took some more classes at the community college actually in computer science to be a better programmer, because at that point I had only learned how to program on my own, which now you guys have a ton of programming resources. So there's really no excuse to not just take a dive and learn today how to write some code. And after that experience, I applied to PhD programs in genetics this time. And I ultimately decided to stay in Oregon, and I did my PhD at Oregon Health and Science University. And I finished that a few years ago. And my PhD work was in cancer genetics. When I talked about like all this tumor evolution stuff, that's what I was doing all day, every day. And I had the honor to work with patient samples, which is they're hard to get, but you get to see like the real impact of your work when you work with the clinical data. And so then, oh, that just went through. I think one of my keys is stuck. That's why I keep accidentally going through more slides than I need to. Yeah, so what I do now is I work in clinical genetic testing, and I work for this company Invitae, which, like Stephanie said, is her competitor. But it's a friendly competition. It's nothing wrong with it. We need more people doing this work. So we offer genetic testing for medical purposes. So we do it if you want to diagnose an unknown disease, or if you're just curious and you want to see if you have any genes that might cause, if you have any variant mutations in genes that might cause or be associated with genes, if you might be a carrier for certain conditions, and you're thinking about having kids and you want to get that checked out. If you are doing IVF, in vitro fertilization, to help reproduce, we do testing on the embryos to make sure their chromosomes are normal. We also do testing on patients with cancer to see if they might be able to use certain types of chemotherapies for their tumors. We do testing to also prescribe drugs for, I'm trying to think of the word, drugs for mental health disorders and things like that too. So there's a whole host of reasons why you would do medical genetic testing, and pretty much for every person in the world, there's a reason why you might want to pursue this. So what I do is I'm actually just now going back to cancer. I used to work for the last almost 2 years. I worked in reproductive health. So I worked on carrier testing, so testing for families that are planning on having kids, or they're just curious about if they're carriers for certain conditions, and also on the IVF and things like that. But for the clinical genetic testing in cancer, I told you that most cancers are due to the somatic mutations, and they fall in this sporadic category. So about anywhere from 60 to 85% of cancers are just from life, random life. Only 10% are heritable, meaning that they have that germline mutation that you can pass down. And then the rest of them are somewhere in between. So we don't really know if there's a specific gene that you have that is in your germline background that's making you have a stronger risk, or if it's something that you share between your family environment, but they're shared among family, but it's not directly related to a gene that we can look for. And in certain types of cancer, that heritability is much, much, much more common. So like in colon cancer, the example we went through, it's the most, it has the strongest germline genetic background, the strongest heritable background of all the cancers. And then breast is next, and then these other ones, gastric, uterine, and so on and so forth. So if you have colon cancer running in your family, you probably want to get tested because it might be a heritable cancer. Same thing with breast cancer. It might be heritable because it's a strong connection between that germline background. And then the benefit to getting cancer tests for patients, cancer genetic tests, is that if you do have cancer, you can it can be identified early before other methods sometimes, and we can watch it, and you might have access to certain medicines or therapies that you wouldn't know otherwise. You might also be able to participate in certain clinical trials based off of your tumor's genetics, or you might be able to have other treatment options that are like surgical, that have nothing to do with drugs. So And these are just other figures, but I won't go over them. And yeah, this is just a slide showing what I was doing for the last almost 2 years. I was doing reproductive health. So I would use the data from our reproductive health assays and inform the greater community through publications and reports and things like that about what we're finding in these tests. And this would be in prenatal screening. So when these are tests on babies before they're born to make sure that they don't have certain chromosomal mutations. And most of them are like Down syndrome or Prader-Willi's, Angelman syndrome. Those are the 3 most common ones. Everything else is like so rare, we rarely even ever see it. Carrier screening. We do some prenatal diagnostics. That means that we can actually do a genetic test when the baby is inside the womb if we think they might have a genetic condition. Those are really rare too, and it's only for a subset of conditions. And then also we have a heart test, a cardio test for women that are pregnant, because there's an increased risk of certain heart conditions developing due to the stress of pregnancy. So we have that stuff too. And that's all the information that I have for you guys. I know it was a ton, and I really appreciate all of your patience. And I hope there was a few pieces of information that will stay with you, the next coming weeks, or just till this afternoon, tomorrow, years. You guys are all welcome to find me. I mean, I don't think you're using LinkedIn now, but maybe at some point in your life you will. I'll put my LinkedIn and my email address in the Slack. So if you want to keep in touch, and if you have questions as you go through your studies, you're always welcome to reach out. Daniel, do you have a question?
You know how cancer is usually caused by cell division? Is it possible for it to be caused by cell multiplication?
Well, I would say that cell multiplication is probably just a, what are you thinking about when you say it's cell multiplication? Let's start there.
Like normal cells constantly making themselves, so then it almost...
Yeah, so
it brings up the massive...
Yeah, so that's actually, so cell multiplication would be a product of getting one of these mutations that would result in cancer. So you would have that cell making a lot of copies, just like this. It would be copying itself out of a normal frequency. Because cells would normal cell division would happen a certain number of times. And I think what you're talking about is if it happens even more than that. So that would already be going down that line of cancer. That's a good question. That's a very great way of thinking about this process.
Thank you.
Yeah, you're welcome. So I think there's a question in the Slack. So is it Linnea or Linnea? I hope I'm not butchering. Yeah, I hope I'm not butchering your name. Linnea. Okay, perfect. Okay. So when a patient uses radiation as a treatment, they may also get cancer. Yes. They absolutely can. And so this is why radiation therapy is used for a subset of cancers. And this is certainly why as we've improved medicine in certain areas, we've realized there are certain treatments that are better for certain things and that are not as good because of the outcomes. And so with radiation, you have to point the laser at a very specific target. And there's radiation oncologists and actually physicists. So if you're really interested in physics and you want to become a medical physicist, this is what you do. You actually work with radiation oncologists to create the coordinates. I have a very high level understanding of this, but you create like the coordinates where the laser would be targeted to. You do all of the math and the modeling because it has to be very precise. And now they've gotten to the point where they've developed these nanoparticles. And you might have heard of these in the news and things. And basically they can take these nanoparticles, which are very, 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 very tiny pieces of sort of like reactive material that they can inject directly into the tumor so that only tissue that is nearby these nanoparticles will get radiation and the normal healthy tissue won't get radiation. So they're working on making this process better, but yes. Oh, and I see some people, they want to be data scientists. Jade, your mom's a data scientist. Well, I'm sure your mom will be a great asset to helping you know more about that. Sharif, oh, that's very nice of you. You said, why is there not cancer in insects? This is a great question. Okay, so it all has to do with the structure and the order of a organism's genetic information as to why they do or do not get cancer. So the other thing too is that I don't know that much about why insects don't get cancer, but I know why elephants don't get cancer. And this is because, and so think of this, elephants live a long time. They live like 70, 80 years. And so if remember we talked about how cancer is a disease of age. They should get cancer. They should get lots of cancer. Dogs live 10 years and get cancer, right? So why don't elephants? So what was recently found out, maybe just, it was within the last 10 years, elephants have more than 2 copies. They have like 4 and 6 copies of those tumor suppressor genes. Remember I told you those tumor suppressor genes are like the brake. So some of these tumor suppressor genes, and namely the main important one is P53. So remember, we've been talking about P53 a couple of times. I didn't give you a deep dive on it, but P53 is a tumor suppressor. And so the reason why it's so commonly mutated in all of these cancers is because it's like the main brake. If you turn off P53, then you don't have a lot stopping you from just growing crazy. Elephants have, I think it's either 4 or 6 copies of the P53 gene. So
I did not know that.
Yeah, it was a random paper, and it was like a veterinary scientist. And you never think a veterinary scientist would completely turn the cancer like cancer world on end, because when it came out, it was like, whoa, like who would have thought?
That is amazing.
Yeah.
I also saw in a video that the bigger an animal is, the less it's affected by cancer, because animals like the blue whale, rhinos, and elephant aren't really affected by cancer, but an animal like a mouse, which is quite small, will be affected by cancer much worse and most likely to die.
Yeah, that's actually a really good point that you made. So that's the other thing too that's interesting about elephants, is that they're really big. But you would actually, if we were really thinking about how cancer evolves, and it's getting all of these mutations, if you're bigger, you have more cells, which means you have more chances for those cells to mutate. So in theory, you would think that bigger animals get more cancer, but you're right. The smaller ones, like mice and any other small animals, they get cancer. We don't see cancer in elephants. We don't see it in whales. We don't see it in big animals. And that means there's something else going on there. I don't know why the size is like the opposite relationship that we would expect, but yeah, there's a lot that we need to study. Can learn a lot from whales and elephants. So
It's
Skylar,
amazing.
Skylar asks, thank you for the compliment. Do you have any other examples of things with high carcinogen levels or things that give high risk of developing cancer? Yes. So we talked about tobacco and we talked about the certain UV rays. The other big things are the like pollution. That's a big one. Any sort of pollution that you can breathe in is probably coming from something that's been burned. And when things burn, you create those free radicals that we had talked about earlier. And you create these hydrolyzed carbons, like all these complicated chemicals, and all those things contribute to mutations. The other things that you can avoid too are things in your diet that can cause more I would say, I don't want you guys to think like, oh, I can't eat these things, but we have found that an increase in certain types of nitrates that are found in how meat is processed and how fermented products are made that are more common in East Asia. So, and the reason why we know this is because we see when people of East Asian descent, when they change their diet from their traditional diet to a westernized diet, we see less gastric and stomach cancers. And we see more gastric and stomach cancers in diets that are high in traditional fermented foods from that part of the world. But it doesn't mean you can't eat those things. It just means, I mean, barbecue, the smoke from barbecue and steaks, like grilling meat at high temperatures, also creates carcinogen. So it's all about moderation. So if you consume things in moderation, it's okay, you know. And then Leander asks, how does chemotherapy help with cancer? So a lot of chemotherapies are specifically, actually, well, I'll tell you this. The oldest chemotherapies, the ones that have been in use for the most amount of time, they literally just kill cells. They're they're not that good at being super specific. And this is why when people are on chemotherapy, they get really sick and their hair falls out and they lose their appetite. Their hair falls out because what chemotherapy does is it targets dividing cells. Cancer, in order to thrive and grow, has to continue to divide. But you still have other cells in your body that are dividing, and your hair cells, your hair follicles, are one of the ones that divide rapidly because it's your hair cycle has like only a few years of a lifespan, so there's some replenishment. So anyway, that's why your hair falls out, because those cells are still dividing pretty quickly. The reason why you lose your appetite and you get a lot of upset stomachs is because the inner lining of your intestinal system, that also those are rapidly dividing cells. It will make you infertile if, like, during that time, if you're male, because sperm are rapidly dividing cells, like they're continuously being made every few weeks. But it doesn't affect your cells that aren't dividing as much. So like your internal organs, like in your brain is not dividing. Those cells aren't dividing anymore. Your heart cells aren't dividing. Your liver cells do still divide, kidney, not so much. So that's how it works. The old way. The new way that chemotherapies they're now designing them is that they're identifying those specific mutations that are really common for particular types of cancer, and they're figuring out how does that mutation change the protein, and then how can we basically make a molecule that can like kind of wedge its way into that protein change and change it back. So it's kind of like they found the protein is like bent because of the mutation in the cancer. So now we need to like wedge something in there. So they make these drugs that are called small molecule chemotherapies, and they can work that way, or they can bind to the cell on the outside and like basically kind of signal like this cell, because I'm binding to like this specific cancer protein on this cell's got to go, you know, and then it can tell the immune system itself to die, or it can like help the cell do what it should be doing and die. So that's kind of chemotherapies at a very high level.
I think Serif Hamed had a question. You can unmute yourself.
Oh, yeah, go ahead.
Hamed? Oh, he put the question on the board.
Okay.
Oh, before Aiden asked, anyone heard of HeLa cells? People say yes. And Hamed says, how many mutations can be together integrated to cause cancer?
Yeah, that's a really good question. And that is literally the money question. We don't know how many mutations are required to cause cancer. But what we do know, and the reason why we can kind of see this is, so what scientists will do is they'll identify the mutations in humans, those cancer mutations, and then what they'll do is they'll go back to cells. They'll do experiments in like petri dishes, and it's called in vitro experiments. So you're doing it in a cell environment, and they'll make those mutations in individual cells and see if they turn into cancer if they just do one mutation. And now they'll do 2 mutations, and they'll do so on and so forth until they get that cell to start to look like a cancer cell. They'll do the same thing once they figure out some good mutations that are actually directly causing the cancer. They'll also take those mutations and put them in mice. And what we found in mouse models is that usually you need at least 2 mutational events to cause minimal cancer. And it's very specific, it will cause very specific cancer. Like it'll cause, like we know it works for kidney cancer, which is the cancer I studied. And I think there's like one other, but I know for kidney cancer, it's one of the easiest examples, because in kidney cancer, there's one gene that is mutated in 90% of all the tumors. And if you take that gene and you completely remove it from mice, all they need is one other mutation in another cancer gene and they'll get cancer. So it doesn't directly answer your question, Sharif, but it gives you an idea that it's not a lot. And Eden, thank you for bringing up the HeLa cells. Yes, if you guys have not heard of HeLa or Henrietta Lacks, I can throw the book into the Slack as well later